Hello everyone. In the last video, I have discussed about the structure and functions of the different parts of the seed found in the fruit of angiospermic plant. Now in this lecture, I am going to talk about the types of the seed. So on the basis of number of cotyledon which is present inside the seed, there are two different types of the seed as dicot seed and monocot seed. Now, dicot seed is generally show presence of two cotyledon inside the seed coat, while monocot seed show single cotyledon. Now, both these type of seeds again divided into two subtype on the basis of presence or absence of endosperm. So, dicot seed further divided into two subtype as non-endospermic also called as Exalbuminous and endospermic or albuminous. So, if the dicot seed show presence of endosperm or nutritive tissue, then such dicot seed is called as endospermic seed or albuminous seed. While if the endosperm is absent, in that case, dicot seed is called non-endospermic or X-albuminous seed. Similar two types is found in monocot seed. If the monocot seed show endosperm, it is called as endospermic seed or albuminous seed, and if the monocot seed do not show endosperm, in that case it is called as non-endospermic or X-albuminous seed. So these are the four different types of seeds found in the fruit of angiospermic plant, and they are on the basis of presence or absence of endosperm. Now in this video I am going to talk about the first type, that is non-endospermic or X-albuminous dicot seed. Okay, so generally, flowers show two different types of essential whorl as androecium and gynoecium, out of which gynoecium is the female reproductive whorl and individual member of gynoecium is called as carpel. And each carpel is made up of three different parts. Okay, suppose we draw a structure of typical carpel found in the flower of angiospermic plant, then we can see three different parts in it. First is stigma. Okay, it is the site where pollen grain fall after pollination and germinate. Then a long tube is attached to the stigma and it is called as style. And the last part of the carpel is ovary. So gynoecium or carpel is made up of three different parts, and inside the ovary one or more ovules are present. Okay. Anatropous ovule means the ovule show opening called micropyle at the downward side, and in many cases in dicot seed generally ovary show anatropous type of ovule. Okay, now before starting with the actual dicot non-endospermic X-albuminous seed, we have to know different parts which is present in the ovule. Okay, now after fertilization, when pollen grain germinate on the stigma and a pollen tube transfer male gamete through style, and when they deliver to the ovule, the fertilization occur inside the ovule, and after fertilization ovary get converted into fruit while ovule develop into seed. Okay, and now the ovule is the only structure which is present inside the ovary that lead to formation of the seed. Ovary only take part in the fruit development process. So before starting with the actual structure of the seed, we have to discuss few things about the ovule. So in ovule, anatropous ovule, the rounded structure is present, and this rounded structure is called as body of the ovule. Okay, so this is the body, and now the ovule is attached to the ovary by a stalk-like structure, and this stalk is called as funicle. In some book, they have written the stalk instead of funicle as funiculus. Okay, so both are same. Funicle means stalk-like structure which is attached to body and wall of the ovary. So this is the portion, okay, which connect body of the ovule to the ovary wall. Okay, and funicle play important role of giving support to the body of the ovule, as well as it provide or it transport nourishment or nutrient from ovary wall to the body. Now this is the main function of the funicle, that is transport of the nutrient. Now the funicle or stalk is attached to the ovary wall at a special point, okay, which having a fertile tissue, it is called as placenta. So placenta is the tissue which help in attachment of the stalk or funicle to the ovary wall. Now the point at which funicle is attached to the main body of the ovule is called as hilum. And if we draw funicle like a solid body or solid part, now when funicle attached to the body, some part of the funicle extended over the body, and this extended part is called as raphe. So these are the three important points that you have to remember in the stalk, that is funicle, then hilum, which is the site of attachment of the funicle to the body, and raphe, it is nothing but the extended portion of the funicle over the body. Now body is the main fertile part of the ovule which is present inside the ovary. The topmost end of the body is called as chalaza. So this end of the body is called chalazal end, and this chalaza is responsible for production of the two layers in the body, and these two layers are called as integuments. So these are nothing but the outer protective covering which is present on the body of the ovule, and this covering is called integuments. Generally, two integuments are present in the typical angiospermic ovule, outer integument and inner integument, and one terminal end of the integument show gap or a space, and this space is called micropyle. Okay, so now we are talking about the body of the ovule, which show three important parts: chalaza, micropyle, and integument. Inside the integument, actual bulky tissue is present, and now this portion which show many haploid parenchymatous cell are called nucellus. Okay, and now inside the nucellus, at micropylar end of the nucellus, a special sac-like structure is present. It is called as embryo sac. This embryo sac show 7-cell, 8-nucleus structure inside it. Okay, so at micropylar end of the embryo sac, 3 cells are present. The central cell is called egg cell, and 2 neighboring cells which surround the egg cell is called synergids. Then at the middle of embryo sac, single cell is present which show 2 nucleus. That means this is the cell which show 2 different nucleus inside single cell, and this structure is called as secondary nucleus. And to the chalazial end of embryo sac, 3 more cells are present, and these cells are called antipodal cells. So now you can see inside the embryo sac, total 7 cells are present, 3 cells on the chalazial end, 3 more cells on the micropylar end, and 1 cell at the middle of the embryo sac. So total 7 cells are present, but now these 3 cells will going to have 3 nucleus. The micropylar 3 cells will going to have 3 haploid nucleus, and the middle cell will going to have 2 nucleus. So total 8 nucleus is present in the embryo sac. So we initially call it as a 7-cell, 8-nucleus structure. Okay, so this is the all about the ovule. Now we will going to see the function of different parts which is present in the ovule. Number 1 is stalk, which include funicle, and I have already told that funicle play important role of attachment of the main body of the ovule to the ovary wall at a placenta. Now these funicle have vascular strands or vascular tissue, so it also transport nourishment from ovary wall to the main body. The point at which funicle attached to the main body is called hilum, and the extended portion of the funicle over the main body is called raphe. Then the main body of the ovule show 2 ends. Number 1, the topmost end is called chalazial end, which produce the outer covering of the ovule called integuments, and this integument leave a space at the terminal end which is called as micropyle. Then inside the integument, nucellus or bulky tissue is present, which is nothing but the parenchymatous cell, and inside the nucellus a sac of different cells is present called embryo sac. In embryo sac, at the micropylar end, egg cell is present and 2 synergid cells are present. Now this egg cell is a haploid cell which will going to produce embryo after fertilization with the male gamete. Now the central cell is called secondary cell, and this secondary cell will going to produce endosperm after fertilization with the secondary male gamete. And now these antipodal cells, they are degenerate after fertilization process. That means they are accessory cells. Okay, so this is all about the ovule and function of different parts of the ovule. Now we will going to see how exactly this ovule which is present inside the ovary get converted into seed after fertilization. Okay, we are talking about the dicot seed in which albuminous subtype. Okay, so dicot exalbuminous subtype. Dicot exalbuminous or non-endospermic seed. Suppose we draw a bean-shaped structure, which is a kidney-shaped seed found in the beans. There are various examples of the dicot non-endospermic exalbuminous seed like sunflower, pea, beans, mango seed. Okay, suppose we draw a bean seed here. Now, after pollen grain fall on the stigma, it germinate and pollen tube transfer from style and reach to the egg cell through micropylar opening, and once it reaches to the egg cell, it delivers male gamete to the egg cell. Now the male gamete fuse with the egg, and in this point or at this point fertilization occur, and after fertilization the integuments or the outer covering of the ovule get converted into seed coat. Okay, so the first part in this type of seed is seed coat, which is nothing but the modified integuments of the ovule, and two seed coats are produced. The outer seed coat, which is hard and resistant, is called as testa. This outer seed coat is produced from outer integuments. Then inner seed coat is thin and membranous called tegmen. This inner seed coat is produced from inner integuments after fertilization. So the seed coat is nothing but the modified integument in the dicot non-endospermic seed. Now these two seed coat play important role of protection of the seed embryo from the environmental factors. Okay. Now inside the seed coat actually embryo is present in the form of embryonal axis. Okay. When male gamete fuse with the egg, then embryo is produced, and inside the seed, dicot seed, it is present in the form of embryonal axis. This embryonal axis is also called as tigellum. Okay, so you can call embryo as embryonal axis or tigellum when it is present in the dicot exalbuminous seed. Now this embryonal axis show two cotyledons attached to it. And these cotyledons play important role of storing the nourishment or nutritive tissues. Okay, so this is the site that store the nutrient, and it will going to provide the nutrient to the developing embryo at the time of germination of seed. Okay, now the embryonal axis show different parts in the seed. The part which is towards the chalazial end of the ovule is called as plumule, and part of the embryo axis towards the micropylar end is called as radicle. So these two are the important point which is present in the embryonal axis or tigellum. The plumule is the part which will going to give rise to shoot system after germination of this seed. When seed germinate, plumule will going to produce shoot system or stem. Then radicle will going to produce root system after germination. Okay, water come inside the seed through micropyle, and once enter the enzymatic process begin, and radicle come out through this micropyle and produce root system. Now the point at which cotyledons are attached to the embryonal axis is called cotyledonary node. And the region of the embryonal axis which is present in between plumule and cotyledonary node, this region is called as epicotyle. Epi means above and cotyles mean cotyledon. That means it is present above the point of attachment of cotyledon on the embryonal axis. Now the region of the cotyledon embryonal axis which is present in between cotyledonary node and radicle, okay, this portion is called as hypocotyle. Okay, so embryonal axis show 5 different points. Number one, plumule towards chalazal end, epicotyle, then cotyledonary node, hypocotyle, hypo means below, cotyle means cotyledon, and radical. Okay, and cotyledons are attached to the cotyledonary node. Remember that this is the Exalbuminous seed. So this type of seed do not show endosperm or storage of the nutrient. Directly, cotyledons store nutrients. Okay, so in this type of seed, cotyledon become thick and fleshy. Now, if the seed do not have endosperm, in that case, cotyledon play a secondary role of providing nourishment to the embryo. Then you have to remember that it is a dicot seed. So I have drawn two cotyledons here. If it is a monocot, then we have to show single cotyledon attached to the embryonal axis. Now, the concave site of the bean seed, if you observe carefully, in that case, you can find three different points at concave site. The first mark, which is easily visible on the concave site of the seed, is nothing but the micropyle end or micropyle of the ovule. So you can see this micropyle on the seed at concave site. Then the middle scar which is present on the seed is nothing but the point which showing hilum. Okay, so it is hilum. And the next or the last scar which is present at the topmost position on the concave site, which is nothing but the rape. So it is, okay, so 3 point on the concave site sequentially represent micropyle, hilum, and rape of the ovule. So in this video, we have discussed in detail about the dicot non-endospermic Exalbuminous seed, which is found in the plant like sunflower, pea, beans, mango. First we see the carpel, structure of the carpel. Inside the ovule is present, and after fertilization, ovule get modified to produce dicot seed, which is without endosperm. So this is all about the first type of seed. In the next video, I'm going to talk about the dicot endospermic seed or dicot albuminous seed. Thank you.